pleasure to invite first speaker of this session, Alfredo Mari, who's gonna reveal to us his journey to his North Star. The adventure started at Max Planck Institute, where he obtained his Ph.D. And then Alfredo pursued his career in Basel during the time when the SARS-CoV-2 pandemics, which you all remember, struck. And it's also part of the story. And so far, the path took him to SOPHiA Genetics, where he works on bioinformatic product development. So, Alfredo, please share your journey with us.
Thank you, Dennis. Thanks a lot. So thanks for the nice introduction. So, I would like to bring you through the journey to my North Star. First of all, let me thank the organizer of this great event to having given us, SOPHiA and me myself, the possibility to bring my career path to your audience. And then, so without further ado, my name is Alfredo Mari, and I will tell you about where I come from and what was, let's say, the leitmotif of my career. So, I come from a different place than what SOPHiA is actually dealing with. SOPHiA, you will see that in a second, deals mostly with rare disease and cancer. I come from a very different field. But though the field in my career changed quite a bit, there was always one main direction, one main North Star, and it was this. So how can I make my work count? So like, how can I make the difference? I am a computational biologist. How can I make my work impact as many people as possible? So with that in mind, perhaps is a least obvious conclusion, I started my career with a PhD in which I was not focusing at all on cancer or human biology. I was actually sampling plants around Europe, and I was studying the microbiome, the microbiology on the plant surface to try to understand what interaction leads to what biological mechanisms on the plant surface. So, as I see many of you here are actually doing their PhD, what is that you care inside your PhD, I guess? Results. So when will you have your results that hopefully you can place on a nice paper, have your publication, and hopefully move on? So as all of you, I was focusing on the result. I was trying to find any possible mechanism that was interesting to find, that would have an impact, but also that could be published nicely. So though making an impact was a very important thing to me, and for this reason, during my postdoc, I actually said, okay, how about using my skills in microbiology, network analysis, not just on the plant surface, which could develop into a nice crop in 20 years, possibly, but how about using them right here, right now, on the health of patients? And together with my professor in the University of Basel and University Hospital, we were actually trying to find a way to find good bacteria displacing bad bacteria. So that was a nice project, still focused on the results. However, when I joined the University of Basel and University Hospital in the virology and bacteriology section of the hospital, this was in October 2019, so the timing was perfect, because basically few months after, unfortunately, SARS-CoV-2 struck in everybody's life. And as you can imagine, everything got sidetracked. So like the good bacteria, bad bacteria, yes, it's nice, it's important, but this stays on the side. So in that matter, so we were tasked, since we were a capacity of the University Hospital of Basel, we don't want to now find results. We want to have a process, actually a pipeline, that in an accurate, reliable, flexible, and portable way could input sequencing data and output variants of SARS-CoV-2. Why this was important? We actually managed to do that because through such a pipeline we could track SARS-CoV-2, so the epidemiologics of SARS-CoV-2 in Basel and in Switzerland, it was actually done in a cool way so that it became actually a reference for other hospitals as well, and enabled actually to share the data and track even more the evolution of SARS-CoV-2 in a very fast-paced, as you all remember, time of our life. And also supported 3 publications. So this was what made me think for a moment to actually say, okay, this is the first time that I focus actually not on just on results to publish, but actually on a process, on a robust and solid process. And this is actually not only is making an impact, but is also getting the results that I always wanted. So this was actually putting myself closer to SOPHiA, because what I was thinking at the time was like, okay, that was nice to do. How about doing that full time? How about doing that as a job? So that's where I got to know SOPHiA, a company that was founded not so long ago, but we are already in 72 countries. We connect more than 750 healthcare institutions. We analyzed almost 1.5 million genomic profiles, and we feature in almost 500 publications from the foundation until 2022. But as somebody in the audience asked me, so what is that we do? So what is that SOPHiA is doing? SOPHiA is offering tools for genomic profiles, for genomic profiling, so is enabling our healthcare clients to analyze the data, genotype the data, and own the data. But what makes us special? Besides that, we are also fostering the decentralization and also the multimodality through our platform, meaning that, yes, we offer algorithms, but we also offer a platform in which healthcare institution and medical doctor can own their data, can interpret their data, and can combine them also with radiomics data and imaging data as well. So, as I was mentioning, our clients are in general university hospital, cancer center, specialty labs. This is because we are mostly focused on rare disease and oncology. Any institution that has basically an in-house sequencing facility, what we offer is our platform and our algorithms to the client side, and they use it to analyze their data, they interpret it, and then these insights that they get out of the data can go either into research projects or in diagnosis. So, but what is that my contribution within SOPHiA? Well, 3 words would be that we explore, create, and improve. I am part of the pipeline development team, so I am really inside, let's say, the data science and where the pipelines get made. But actually, to say with the words of my boss, which is the king of analogies, I would say, so we do not design engines or wheels, but we build trains. I will come to the trains for a moment. Just let me share with you one example of exploring and create products and pipelines. So I don't know how many of you are actually knowledgeable on gene fusions, but just so that you know, when you have 2 different genes, you would expect that these exons within the genes would rearrange between themselves. But in very aggressive cancer types, you actually have that this part of genes venture with others, and this is what you call a gene fusion. This is a sign of a very, very aggressive cancer types. So one client came to us saying, look, I want one of your products, but I just want to detect gene fusions in this specific pool of genes. And it was a pool of genes in which it was rather difficult to detect fusions. So we built it, so we created, we analyzed it, we tested it, and then we submitted again to our client, which was saying, there is something fishy here, because we are continuously finding this gene fusion here, gene A, gene B, gene A, gene B, and this is not reported in literature. So what we did was to actually plan further experiments together with our customers to see whether it was our pipeline that was confounding noise with actually true biological signal. but it wasn't the case. So it looked like our pipeline was catching true biological signals out of these samples, and now we have a publication underway. So this is a case, for example, in which we do actually foster research. But let me come back to the nice, let's say, train analogy of my team leader. So I told you that we build trains. So what do you need in a train? You need first a railway. So you need an infrastructure that is able to run your pipelines, your train, faster and safely till the destination. This is what our tech department and software engineering department is doing. Then also you need experts into the actual wagon mechanics, vertical experts that know everything about the wheels, about the brakes. And this is, for example, our algorithm development department. But ultimately, you need somebody that is making the train happen, that is making the right train for the right purpose and ensures that this train arrives to the destination. And this is the pipeline development team of which I'm a proud member. So why the purpose and why the oversight is important? Because as you can imagine, there are different kind of pipelines, different kind of trains for different purposes. And even if you have the latest wheels of the next generation, but you want to move people from Ushi to Crozet with a cargo train, that's missing the purpose. Nobody would need such a transportation means. So this is what we are actually doing. So getting more into the detail, I want to give you one example instead of what we do when I tell you improve what we have. So once we create something, then we improve it. So take our example train, which is our pipeline divided into modules and sub-modules. Typically, the input and output flows from one to the next, to the next, to the next. We are making sure we are the gatekeepers of best coding practices. So like if somebody does codes not in a, let's say, obscure way, we're actually saying, no, you shouldn't do that. But one improvement, for example, that I was involved first person was we had one old pipeline that had many modules, that had like over 40 modules. And actually, the goal was like, this pipeline is stuffy, it's slow, it produces many things that are not necessarily needed. Can we make it shorter? Can we make it faster? Because a shorter train runs faster. And it turns out that actually we could shrink it almost to half. And into another project, for example, see what we have here. We have 2 different pipelines that are using 5 different modules. What happens if you have all of this living in the same place, stored in the same place? You want to change something in module 1 for pipeline 2? Well, if this module is also used here, you may break something when you run this pipeline there. What is a solution? A solution is give each module their own little house, so that when you call pipeline 1, you call one version of module 1, and pipeline 2, a version of module 2, the version 2 of module 1. So why I'm telling you this? Because if you couple these improvements all together, you have increased trackability, improved maintainability, and faster processing. Now, this happened for 5 modules and 2 pipelines. We have over 300 pipelines, and every pipeline is typically more than 30 modules strong. So when you improve such a thing, and you multiply by all the customers that we have, in all the institutions that we have, this means a diagnosis that can be achieved earlier, means a feature that can be added faster, means research projects that can be delivered earlier. So this already gave me a good idea of how can I make my work count, but in general, I want to leave you with 3 main key messages. So what we are doing in the pipeline development team is that we are detail-oriented, because sure, we are not the algorithms guy, but we need to know if there is a weird noise in our train, we need to know why is this. We don't just start pipeline and cross the fingers, hoping that everything is good. That's not how we approach things. We want to know everything till the end. But also, we need to have in mind the large scope that we are fulfilling, because, again, we need to focus on the trains, not on the single part. And also, of course, we need to be proactive, because once the train is stuck, that's too late. We need to anticipate breakage, anticipate problems, and make problems not to happen in the first place. Now, what I was telling you at the very beginning, my goal was to make my work count. So the example that I showed you about the gene fusion was the RNA target oncology solution. The improvements of the two pipelines was the whole exome solution and clinical exome solutions. And these are figures of how many genomes were analyzed in 2023 by just the 3 products that I touched. So when I saw this figure, I thought, look, my work was actually used for almost 40,000 patient data on which now medical doctors have insights on. This is what I would call make my work count. With this, I'd like to thank you, and very happy then to take questions and discuss with you at the booth.